Okay, we're live.
We are live. That's beautiful. Thank you, Olga. Warmest greetings. I'm Chris, the CEO of Barkey, coming to you live from Germany with my co-host Olga, the manager of Aseptic Technologies, live from Belgium. This is a new edition of the Insights with Chris and Olga, the Cell and Gene Talk, a webcast where we interview a different panel of experts in the cell and gene therapy space to keep you updated. Hi, Olga.
Hi, thank you, Chris. Hello and welcome. So today we have a very special topic. It is not about COVID-19 and it's not really about manufacturing of the cell and gene therapies. It is first of all about personalities bringing the revolutionary therapies to the world. And today we talk about Dr. Carl June and Emily Whitehead Fateful Meeting. And it's a story of dedication, belief, and changing the paradigm of healthcare.
Truly inspiring, Olga. Thank you. And let's welcome our today's guests. Olga and me, we are really, truly honored to meet legendary Dr. Carl June, director of the Center of Cellular Immunotherapies at the Perelman School of Medicine and director of the Parker Institute for Cancer Immunotherapy at the University of Pennsylvania. Hi, Carl, and welcome.
Hi, Chris. It's really a joy, and Olga also, to join you on this occasion, really, to talk with Tom Whitehead and his daughter Emily, and the wonderful journey we have had over the last almost 10 years now.
Great. Thank you so much, Carl, and welcome. So we're also delighted to have with us today Tom Whitehead. He is the co-founder of the Whitehead Foundation and father of Emily Whitehead, who was the first pediatric patient that was treated with the CAR-T cell therapy. So warm welcome, Tom. How are you?
Thank you very much. It's like Carl said, it's an honor to be on here today, and every day I feel blessed to still be Emily's dad, and we try every day to do something to help more families have the same outcome as us.
Great. Thank you so much, Tom. So we're about to start, and stay with us to meet Emily, that will join us by the end of the show. Also, by the end of the discussion, we will have a short Q&A session. So don't hesitate to comment below to introduce yourself and submit your questions so we could pick some of them for our discussion. So let's start. Chris, why don't you start with your first question?
Yeah, thank you, Olga. Tom, 9 years ago, Emily's acute lymphoblastic leukemia relapsed twice, and you were basically told the doctor had run out of options. Can you describe the moment when you learned about the CT19 that later was named Kymriah study at the CHOP, and how did this news make you feel?
Yeah, so when Emily was diagnosed, we started treating at the Penn State Hershey Children's Medical Center there in Hershey, Pennsylvania, which was 2 hours from our house. We live in Phillipsburg, Pennsylvania. And when we got started, we were told, that the acute lymphoblastic leukemia is the most curable kind of childhood cancer. It's the garden variety kind of cancer. If you have to have a kid with cancer, this is the one you want to have. And if we just followed the protocol for 26 months, they told Emily, you'll grow up and become a grandmother someday and you'll be fine. She had a very rough start, just after starting chemotherapy in the induction phase at the beginning. She developed leg infections and almost had to have both of her legs amputated. She went into a surgery where we thought that she might lose her legs. So they saved her legs, and she stayed in the hospital that first month, but the treatment actually was working. So we got to take her home. She got started there on May 28th of 2010. And we took her home in remission after the first, 6 weeks or so. And we thought we were going to get on track and have it go that way. And then 16 months in, while still in treatment, she relapsed. And when we found that out and they told us, now she had less than a 30% chance of survival because kids don't usually relapse in treatment, we decided to go for a second opinion before we moved forward with a bone marrow transplant. And we went down to the Children's Hospital of Philadelphia. And that's when they said, at that time, bone marrow transplant was the best option. But they had mentioned that there was an up-and-coming T-cell trial that they told us about that we looked into, but we didn't think Emily could survive long enough to get into that trial because they thought the FDA was still months away from giving them permission. So we tried to get the bone marrow transplant back at Hershey. And during that time, we read about Carl and the first 3 adults that he had treated there at Penn. And we studied, about Carl's research. And my aunt was a medical student at Hershey, studying to become a doctor. And Carl's son was there with her. And I'll never forget that she said to me, hey, you got to look into that Dr. June. His son is the smartest guy in my class. So we felt strongly that that's the way we wanted to head if we could make it that long. But then finally, Emily relapsed again back at Hershey, and they advised us to go home on hospice and enjoy the days we had left. And I had felt strongly inside all along that she was going to get better down at CHOP there in Philadelphia. So when we paged Dr. Reingold saying, we're bringing Emily no matter what you have, she said, well, I find this amazing that you paged me today, because yesterday the last email I read was the T-cell trial opened months ahead of when we expected, and we think Emily would be a great candidate to become the first patient to receive it. So we transferred down there on March 1st of 2012. We were able to transfer down there, entered the trial, and then they took Emily's cells and took it over to Bruce Levine's lab there at Penn to grow them. And then she stayed in isolation in her hospital room for 6 weeks until they were ready. And then she got them back on April 17th, 18th, and 19th of 2012.
Right.
This is really incredible. Yeah.
Yeah. Well,
thanks so much.
Thank you. Yeah, thank you, Tom, for sharing this. Okay, so then the next question we would like to address to Carl. So, well, it took over 20 years to develop and then make available the same treatment that was provided to Emily back in the days. So when did you realize that you and Emily had created a whole new paradigm with the treatment of cancer?
Well, Olga, it was one of those like an up and down thing. So we had treated, as Tom mentioned, many mice that had leukemia, and we showed that we could cure leukemia in mice. And then we treated 3 old men with a different kind of leukemia, and 2 out of those 3 people now, 10 years later, seem to be cured. And so they had a very good response. But when we treated Emily, we had something that we had never seen. As Tom mentioned, in April of 2012, the cells were given on 3 different days via IV infusion, like a blood transfusion. And on the 3rd day, then she became very, very sick. She had a very high fever. It was almost 41 degrees. And it was not an infection. And then she developed multi-organ failure. And something we later found out was called cytokine storm. And so she became comatose, was in the intensive care unit. We're really very close to death. And we found out she had a very high level of a blood protein that causes high fevers. It's called interleukin 6. It was over 1,000 times above normal. And fortunately, about 6 months before that, in the US, the FDA had approved a drug that counteracts the effects of interleukin 6 and causing fever. And so her physician at that time, Dr. Grupp, we told him we had found this very high level in the blood, and maybe that's why she was having the high fevers and the multi-organ failure. And so he got approval from Tom and Emily's mother, Carrie, to give this experimental drug to her, because it was made for arthritis. And we gave it in what's called off-label. So off-label use for her cytokine storm. And it was really remarkable and miraculous what happened.
Right. In what in everything that you and Tom are saying, I can see that there is a lot of, you know, everything is about the right timing in this story. That's really impressive. And maybe a short follow-up question here. So for you, Carl, what would you define as the most important milestones in your research and the research of your group that preceded the CT19 and what is now called Kymriah study?
Well, thanks, Olga. So, we worked on it for about 25 years. And there were a lot of times in science, most times experiments fail. And I always make the analogy to baseball, where in baseball, even the best players in the world only get a hit about 1 out of 3 times. So 2 out of 3 times they fail. And you have to get used to failure. And so we had many, many failures. But one of the most unexpected was, I was trained as a leukemia doctor. I spent 3 years in Seattle at the University of Washington doing bone marrow transplants and learning leukemia therapy that way. But then I was in the Navy and I was not allowed to do leukemia research. And so for 10 years, I had to work on HIV, which was an epidemic just coming out in the 80s and 90s. So by working on HIV, it was actually a very unexpected blessing for making CAR T cells for Emily, because we used an HIV virus that we modified to insert the gene for the CAR molecule into her T cells. And so if we had not done HIV research, we would not have known how to use the gene technology for Emily's CAR T cells.
Absolutely. Great. Thank you. Thank you, Carl, for this comment. Chris, would you like to continue?
Oh, yeah. Thank you so much, Carl and Tom. Starting with Carl, because you just mentioned it's all about failure. What motivates you?
So that's a really good question. It's Chris, there's a lot of motivation, to help someone else is probably the best thing you can do for yourself even. So, pediatric cancer patients it's clearly, it's a huge unmet need when, most people with relapsed leukemia die and in a very short time. My first wife got ovarian cancer when she was 41 years old and died about 5 years later. And that was very frustrating to me because I'm trained as an oncologist and we could not stop. She got all kinds of chemotherapy, very toxic, bone marrow transplant, and still, she passed away. And so that gave me a big motivation, with my laboratory colleagues like Bruce Levine, who made Emily cells that you heard Tom described. It gave us a lot of motivation to actually not just do mouse experiments. It's a lot harder to take them out of the mouse and then do them in humans. And that's what we had a lot of motivation to do.
Yeah.
Thank you so much. That's really, really touching. Tom, for you, the same question doing what you do. And when we met the very first time, you told me also about your very, very strong belief, which I was really as well impressed about. What motivates you and, of course, the whole family now?
Yeah, so I think, when Emily was in treatment, there was a lot of other parents there fighting right along with us, fighting just as hard as us to get their children better, and they lost their children. So I think, thinking about that and witnessing that, it was brutal to watch that happen. And then some of those same parents that lost their children even jumped in and made suggestions to us to help us have success. And I think, seeing what they did and knowing how much pain they were in, but they still reached out and helped us, and I can tell you when Emily was really sick and in that coma, personally, I was doing a lot of praying and saying that, if Emily comes out of this, I will do everything I can in the future to help other families have the same, outcome as us. So, I mean, since Emily got better, we never expected everything that's happened. But I feel, responsible to do everything we can to help other patients get the treatment and make it accessible globally. Because when the international media attention came to Emily's story, so did a lot of calls from parents from all over the world saying, please help us have the same outcome as you did because our child's being sent home to hospice. So, I had what I called some whispers that guided us. And Carl told me afterward, he said, whatever guided you to make at least half a dozen decisions that led to Emily's success, to me, that was a miracle because any one of those you change, Emily wouldn't be with us. And we definitely, had guidance there. I did. And we're going to write about that in our book. But I just felt that's the path I was put on. And with Carl spending his life trying to make it happen for Emily, I just thought I'd like to walk that path the rest of my life to help bring in the funding needed to help more kids and just do whatever we can to make a difference in these other families' lives that still have kids fighting cancer.
Really great. Thank you, Tom. It's a really great mission and a very strong statement. So can you now describe us what is your daily routine? What are you doing in your foundation and also within your private Facebook group? Can you share with us what's happening now?
Yeah, so when we went through that, we didn't have anybody to turn to as far as people with experience because Emily was the first in the world. So once we got done, we got with some of the other families that went through it early on, and we started a private Facebook page for the parents or of the patients, sometimes just the patients on there that are older. So they had, access to get information from people who lived it before them. So while you're going through, if you experience a cytokine storm, you can get online and ask questions and get answers from parents that already have that experience. So I think we're very proud that we have over 300 families on there, I believe. And it's been like a close-knit group of people who didn't want to be part of the group, but now are very, they also want to make a difference too and help others once they've lived through it. And it's, some of those parents didn't have a good outcome too, but they're still on there to help others to try to have success. So I think with that, helping with that group and then with our foundation, we realized a lot of parents can't figure out how to find these clinical trials. So we we did a lot of work and made it easier. My wife has a lot of research background and science background, and she wanted to create, with a lot of help, search engine to help parents who don't have science background be able to find an open trial instead of going home on hospice. And then we're out all the time, going to conferences, spreading awareness, talking to Congress members, trying to get more funding, and just doing anything we can to make a difference to help more people survive cancer.
Great. Thank you, Tom, for sharing it.
Yeah, thanks.
Really important. Yeah, so Chris, would you like to continue?
Yeah, I just want to let you all know, Bruce is just sending out a message and is sending a comment, Carl and Tom, Science and Serendipity, the amazing story of Emily Whitehead and the development of CAR T cells. Is he just posting? The question, Carl, to you is the research and development of Kymriah, where you're the co-inventor, occurred almost entirely at an academic center, the University of Pennsylvania, which is highly unusual. Can you tell us more about the main challenges you and your team faced and the role of media and communication in raising awareness and access funding?
Thanks, Chris. Yeah, it was, I think, something that no one thought would ever happen, where you would actually make a drug from the patient's own cells. I mean, we've had and talked about in medicine, initially all of the therapies for cancer are basically cell poisons, and what we call chemotherapy. And so those are all made, and they're given to people, and you don't know if it will work or not, but everyone does have toxicity from, throwing up and their hair coming out and so on. So people have been used to that in cancer, but they never had a cell that was precision guided and made from their own body. And so what we were able to do with CAR T cells is train them so that they will kill the cancer cells. So by taking the cells out of the patient's body in about 10 days, when we give them back, they're killers of cancer cells, and they actually divide in the patient. So at the time Emily had her cytokine storm, she had thousands of more cells in her than we had given to her just a few days before. And now, almost since 2012, 8 years later, she still has these cells in her. And they're on patrol, and we can show that if you take them back out of her arm by just a little blood draw, they can still kill leukemia cells. So there's never been a drug that's so-called, lifelong effects. Usually the drugs you give them, they go away, they're metabolized. And so that's been a dream, to have a drug that would last and stay on patrol to prevent leukemia and other cancers from coming back. And I can tell you, quite frankly, one of the major challenges was that there was no industry to do this. All the drug companies are really interested only in making so-called, drugs that would treat everyone, and you can make enough for 1,000 patients at a time. They didn't want to get into the complexities of doing what we did, which is more like in the Red Cross, where you get a blood donation and you may donate your own blood and then get it back later. And so that's been a big paradigm shift for the pharmaceutical industry. They are now doing this worldwide. They have different manufacturing centers, and they use airplane shipments, and they can, from each patient, make their own CAR T cells. And this is going to get better and better, the process. It's a huge industry now, evolving. So that was a paradigm shift. The fact that it took just one patient like Emily made the massive pharmaceutical company, Novartis, say, this is something we want to do. It was a complete risk for them. It was because they'd never done a personalized therapy like that. But the results were so striking, they said, well, how can we not do this? So we had an alliance that we signed with Novartis in late 2012. And that resulted then in the FDA approval of CAR T cells in 2017 for pediatric leukemia patients age 3 to age 25. And that was a revolution in medical history of cancer therapy.
Absolutely.
Incredible.
Yeah, inspiring, really.
Olga?
Thank you. Yeah, thank you, Carl. Thank you, Chris. Well, my next question is actually addressed to Tom. So you briefly mentioned that you're writing a book. So indeed, we heard that a book, Praying for Emily, the Faith, Science and Miracles That Saved Our Daughter, will soon be released. I also heard that there is a documentary you're also working on. So in these lights, can you speak more about the educational and awareness missions of the foundation that you already brought in your previous speech, and also your experience in delivering these messages?
Yeah, we've had a complete education since Emily got better in how film and TV works, and now writing a book, and we put a lot of years into it. I think in the beginning, when we were just sharing her story, the science part of our story, and our goal was sharing that and bringing more awareness to that will lead to an FDA approval, which then will make it accessible to many more patients because other countries started passing it. So the first 5 years, we concentrated mainly on just sharing that part of our story. And, in the beginning, Oscar-winning film director Ross Kaufman made a short version of our story with Carl and us, and it went online, and it's called Fire with Fire. Yeah. And it went viral, a few months after Emily Emily's story hit the media. And since then, I think he said it's been viewed over 20 million times. So we saw the potential there with that awareness. And we started working on a full-length documentary, more concentrated on the science part of it. And Carl and Dr. Levine and them are absolutely, we can't wait till it comes out, and it's supposed to come out in 2021. The goal is hopefully to try to apply it at Sundance and get it in there. And we've just seen when we do media, if we do a short film or if we do a longer film and more people find out about it, a few months later, I'll get a call or I'll get a message that says, because your daughter was in the media, because you guys are willing to share your story, it saved my child's life. So, that's pretty powerful right there. But with our book, after the FDA approval came, I had talked to Carl and talked to Dr. Levine and them and said, there's parts of our story that are really hard to explain. And I had some guidance. I had like a vision of Emily getting better before she did, and it led us back and forth. And that's when Carl said to me, he said, I can count six of those decisions. If you change any one of them, she's not here. And twice we went against what the doctor suggested, and it saved her life both times. So that's kind of what I wanted to share in the book. So when Emily got very sick, even from the beginning, my wife started a blog, and we called it Praying for Emily. So I think it was Prayers for Emily on Facebook, but our book is Praying for Emily. And we ended up with 29,000 people on that, sending prayers. And those that don't pray said they were sending positive thoughts or helping us in any way they could with fundraisers. So I think, with the book, we thought that part of the awareness of the story, I wanted to share what happened there and let everyone make up their mind what guided us. So, that's been our goal all along, and it's taken years to bring the book about, and we're just finishing up the final edits in the book. So it's up for pre-sale right now on Amazon, and it'll be on bookshelves in the bookstores on October 6th. But you can order it now, and then it would be delivered to you on October 6th. So the documentary is going to be called Trial by Fire, kind of a follow-up to our short film that was Fire with Fire. So that'll be Trial by Fire. And the book is Praying for Emily, the faith, science, and miracles that saved our daughter. And then I cover more of the faith part of it behind it. And like I said, I'm hoping it'll start a conversation that you don't have to separate faith and science. It all works together. And hopefully it brings a conversation together of people of faith with maybe people who don't have faith. Because I've received hundreds of messages, saying that following your story the whole way through gave me my faith back. So I just felt strongly that I wanted to share more details of that part of our story in the book, because that really hasn't been shared much in the media so far.
Yeah, really interesting how you combine, how both parts, the book and the documentary, will complete one another to really reflect your story and share it with as much people as possible. That's really great. So my next question is for Carl. So slightly different topic. So it's about the fact that just last week, the World Health Organization warned about COVID-19 pandemic being accelerated globally. And I know that you have spoken about being committed to using your expertise in the field of the immune system to support the research of the COVID-19 treatments. So can you please talk more about this, Carl?
Sure. Olga, there's been data that, other coronaviruses that, like one was called SARS, which is very close to the current virus we have, that caused some of the people that died from that virus with a pneumonia also had an exaggerated immune response. And that contributes to the pneumonia and to some people actually dying. And so there's emerging data that happens to a subset of patients with COVID. And we have a trial now testing, and there's probably 50 around the world, testing whether dampening the immune system in some of the patients will improve their survival. So viruses can cause cytokine storms as CAR T cells can, although they're usually not as pronounced and striking as the ones with CAR T cells. But just last week, I got an email from the mother of a 12-year-old patient who was treated with Kymriah. So her daughter had had leukemia a year and a half ago, was treated, and now she's in remission. And her big question is, should she go back to school? And we don't yet know if kids after CAR T cells are more susceptible or not to the virus, if they would get COVID, and whether she should be in school or not. And the American Academy of Pediatrics has recommended that, if possible, all kids go back to school this fall because of the social issues of learning, being in contact with other kids.
Yeah.
So far, I've asked all the pediatric physicians, such as Dr. Grupp and others who've seen more than any other patients in the world. They have not seen that these children like Emily, who are in remission and probably cured, but still have CAR T cells, they do not appear to be more susceptible to the COVID virus.
Right. A really interesting aspect that we really didn't cover that much. Thank you for these insights. So, Chris, would you like to continue?
Oh, yeah. Carl, your fascination with T cells and your research went from cancer to HIV and back, as we learned today. And talking about revolutionary therapies, what are your insights on the development of the HIV therapy in this regard?
So, HIV is a virus that right now can be suppressed but not cured. There's one famous patient called the Berlin patient who actually, Tim Brown's his name, but he was given a bone marrow transplant in Berlin from an unrelated donor because he got a leukemia, and they found that it actually cured his HIV. And there's now been another patient in London where that's happened. So there is now an emerging field of HIV research so that to use cell and gene therapies to actually eradicate the virus from the patient so they don't have to take medicines every day. If you look in textbooks, it will say that retroviruses, of which is what the HIV virus is, it says they're incurable, that once you get one, it'll always be in your body.
Yeah.
But I think cell and gene therapy offers now the potential to actually make a virologic cure in some patients. So there's exciting new research there. And basically this is one of these things of a virtuous cycle. It's, we learned from HIV how to genetically modify Emily and make her have CAR T cells. And now that research and what we've learned, and the industry that the pharmaceutical companies like Novartis have made to grow cells, have made it possible now that that basic technology could come back to chronic infections and be used to treat that way. So I think we're going to see the cancer, research benefit chronic infections like HIV, malaria, and hepatitis.
Right. And great. So probably a follow-up question, really very logic after what you have just said. What are other new technologies and new approaches in the field of cell and gene therapies that you, Carl, are personally the most excited about? Can you share this with us?
So, I'm really excited about the intersection of gene editing and cell therapies. So, technologies like CRISPR allow us now to find a single gene. It's literally the needle in a haystack, and change it so that we can try to make cells better than they have evolved in us. So we can make super killer CAR T cells, I think, that are better than the ones we actually have now, or more potent, and more controllable, so that maybe we won't have cytokine storms. So I'm very excited about that intersection of gene editing, and this is an area called synthetic biology. Another aspect is really engineering, and it's, can we make cells, from, like in blood transfusion technology, we have if you're an O negative blood type, you can be a universal donor. We don't have that for T cells. Unless you have an identical twin, the only T cells you could make that would work in you would be your own. So for people, but a big area now in research is making cells that would come from healthy people and being able to give them to cancer patients. And I'm excited that shows a lot of promise.
Oh, really? Great.
Sounds to me a little bit like convalescent plasma, taking plasma from recovered patient and using this on some kind off-the-shelf product, right, Olga?
Yeah, I would say, it's exactly that, Chris. So antibodies, what protects you with convalescent plasma is the antibodies. And one antibody, if it works in you, it will work in me. That's why we've had monoclonal antibody industry since the 1990s. They don't depend on your genetic background. But T cells, as I mentioned, unless they come from an identical twin, my T cells won't work in you. I happen to be O negative blood, so I'm one of those universal donors. And my antibodies and your antibodies would work in everyone. In fact, Emily gets antibody therapy because she doesn't have B cells now that make antibodies anymore, because that's one of the targets of the CAR T cells. So convalescent plasma works that way because it's derived, from antibodies. And it would also be possible to make T cells that would fight viruses like COVID, but they would have to be matched genetically. So it's a lot more complex than convalescent plasma, where we're going to have pools of that and then be able to give it to patients who are at risk or who actually have COVID.
Thank you so much, Carl. Yeah, absolutely. Olga.
Yeah, so
next question.
Yeah, maybe we can have our, let's say, question that would be totally interesting to see from different positions, from Carl's and also from Tom's. So in your opinion, what are the most important challenges in bringing and providing such revolutionary treatments to the world? And from both, development, manufacturing perspective. So would you like to share your thoughts on that, Tom?
Yeah, and it's been very educational for us to be able to see and watch Novartis build up their factories from the beginning when they got involved and started their cancer factory there in New Jersey. We were able to go and tour behind the scenes and have Emily see it and get to inspire their workers. But that was huge, because in the beginning, when we were getting these calls from families from all over the world, a lot of them couldn't get to Philadelphia. And we could see that for it to become accessible to patients, it needed to be with a company like Novartis. I think Carl told me from the beginning, I need somebody with that infrastructure that can take it global. But we've been able to watch that happen and get the behind the scenes and see how they do it. And it's really blown us away and gives us a lot of hope to see exactly how many people are working on it and how it's working. And, I know of like patients from 11 countries that have been able to get it. But, I think in the beginning, the challenge was very hard for us to hear from parents saying, my child's not going to survive because I just can't get from my country to the United States to get treated. And now those calls, or messages are far less now. But they still do come from some countries where they just can't get to treatment. But we've seen how it's becoming much more accessible around the globe. But our goal is to keep fighting and raising funds and get it so it doesn't matter where you live. If you need the treatment, you're able to get it. And even South America, we've had many messages, and Carl and I have had talks where some of those parents can't get treatment for their kids, and we're just trying to figure out different ways and do whatever we can, even by us traveling to those places and doing some media. Sometimes that puts some pressure for them to invest more into getting that treatment for the people in their country. So we're happy to do that and do whatever it takes to bring that awareness and show them, that our daughter, now leads a normal life because of the treatment, and that can happen for children everywhere. But I think, seeing it become more accessible around the globe is really huge, and we're trying to do everything we can to continue to put ourselves out there and bring that awareness and make that happen for these other families. Sometimes I'll get a message. I had a message from Russia from a 17-year-old girl trying to help save her 5-year-old brother. And, when I was even trying to email them back, sometimes my emails would just bounce back. I couldn't get back in touch with them. It really breaks your heart when you're trying to, to help as many people as possible. But we have many talks with Carl and Bruce Levine and the team, and I try to educate myself so I know if parents reach out to me, I know what the latest upcoming trial is, and make sure that if there's kids out there that need it, that we make them aware of that.
Right. Thank you so much, Tom. And Carl, what's your opinion about these challenges? Can you share with us?
Sure. I mean, there is, with all new technologies, there are usually access issues, and that's not just in healthcare. It happened with cell phones and computers and so on, and then they gradually have been cheaper to make. So one of the biggest problems with cell and gene therapy is they're more expensive to make per patient that's treated. The advantage, as we've mentioned, is you only have to do it once. So in the end, it can be more cost effective. And what we expect is, with all kinds of engineering applications, that it's going to become cheaper and cheaper, the cost of manufacturing, so that it'll have wider access. Bruce Levine and I are working in India, where there's 1.3 billion people, and as you know, it's this Indian subcontinent has no CAR T cell therapies, and there are thousands and thousands of kids every year who get leukemia there and are dying. And there's none of the drug companies have trials in South America or Africa. The only place in the southern hemisphere where there are CAR T cells is Australia. So there are economic disparities. That's true for many things in healthcare, and cell and gene therapy is maybe one of the worst examples of that. It's right now exploding in Europe and in the U.S. And Japan, but we need to get it out, and it's really heartbreaking to get emails from, as Tom mentioned, someone in Russia, and their loved one has a therapy that, if we could get it to them, could save them.
Yeah, absolutely. Thank you for this valuable comment. Okay, shall we take some comments from the audience, or maybe there will be Emily that will come to say hello?
I have Emily and our dog Luna ready to join whenever you'd like her to join.
Yeah, wonderful.
Absolutely, great.
She's coming.
Yeah, Emily had a different dog when we treated her back in 2012.
Yeah, we lost our dog Lucy, but now we have a Chihuahua named Luna.
Luna. Hi, Emily. Hi, Luna.
Hello, Emily. Hi, Luna. How are you?
Good. Yeah.
Great.
How has the lockdown been for you?
Okay. Yeah. Not much to do.
Yeah, right. Great to see you.
Are you going to plan some vacation around, Emily?
I don't know. Maybe.
She really enjoyed our trip to Germany to visit with you last year, Chris. So
yeah.
I think her and Carrie want to come back and see the Christmas markets again in Germany sometime.
You see your masterpiece here, Emily. This is the one you still with us, and the green one will go to Carl later on.
Yeah, we hope to be down there to help deliver it in person, hopefully.
Wonderful. That is our dream, so to say. Emily, any future plans for? I mean, you like to study. I remember that you are in art, and movies is interesting for you. Any updates on this you like to share?
Well, I'm really into film, and I'm going into my sophomore year of high school, so I'm hoping to go to college for film or something in art.
Very good. Any specific
Great.
idea already?
I really like New York City, so maybe college there. I'm looking at that.
Good.
She was
Good
showing
choice,
me,
not too far
she was
away.
showing me the price of NYU Film School. I said, keep studying. We need some scholarships. But we're very proud of her. She's fourth in her class currently, and now she's 5'10". And when we talked about the price of it, the treatment, when I travel around and I'm sharing Emily's story, I said, I think back, and if Emily would have went to bone marrow transplant and had success, I think we would have had far more care since then. And we don't talk about it very often, but Emily's 8 years since she spent a night in the hospital. So when I look back now, from the cost we would have had with our health care had she gone through transplant and all the extra problems that would have came from that, to me, it seems pretty reasonable at where it's priced at now. If you can get it when it works and you're done. We've been very fortunate and just blessed. But to me, but I think it will become more available in other countries, like Carl said, when we can figure out how to bring the price down even more. And we have to be a part of helping make that happen.
Yeah, really amazing. Great message there. Yeah.
So, but when you go to New York, which is not too far from Carl and home, correct?
It's about 4 hours, I think.
Yeah.
About 4 hours.
Carl,
we can get. I was going to say we can get outside the tunnels in about 4 hours, and then it takes us maybe an hour to get to our hotel from there.
Oh yeah. Carl, how far is this from your place where you're living, New York?
And it's on the train. It's like if we would drive, we can take the train and it's much faster. It's an hour and 10 minutes to the downtown train station in New York. So it's very easy to get there.
Yeah. So,
right.
Emily, if you take Luna along, I think Carl will take care sometimes.
Yeah.
Emily got to meet Carl's dog at his office one day. Carl brought his dog into work, and Emily was teaching how to sit.
Yeah, that was Lacey. Yes.
Yeah, it's maybe if I look to the time, it's maybe good to switch over to the Q&A session. But before, I would like, of course, first of all, say thanks to Carl, Tom, Emily, you, and Luna. Of course, thank you so much for being here today, and thanks for sharing your inspiring story and your insights. And I heard we have some really interesting questions. Olga, would you like to start with the one from Christoph Targovsky?
Yeah, I can see it. So we received a question from Christoph, and I guess it's addressed more to Carl. So how do you see the development of the cell therapy industry, manufacturing and logistics in 10 to 20 years? So what are your future visions about the development of the industry?
I think it's hazardous. I can say in the next 3 years, we're going to have much better automated systems. So right now, what we do, what the pharmaceutical companies are doing is pretty much what Bruce Levine invented in my laboratory 20 years ago. And it involves a lot of manual labor by highly trained technicians and scientists. That makes it hard to scale it out to many patients. So when it becomes automated and the cell culture procedures done by robotics, that will make it possible to scale out and probably drop the cost by 50 to 80%, the cost of goods. And I think, so that'll be 3 to 5 years. That's what engineering can do. And then there are things you can't predict that might happen, like that could lower the cost 99%. For instance, we can make so-called iPSCs now, induced pluripotent stem cells, and those could be then using existing cell culture technologies. You could make thousands and thousands of doses from one starting culture. And then the cost would come down dramatically, and it would be similar to pills that you take every day.
Right.
So we don't know how long. There are companies just beginning to test these in academic studies to test so-called universal donor cells, which radically changes the logistics. So I think in the long run, in 10 to 20 years, in reply to what Christoph asked, I think we're going to have both universal off-the-shelf cells that any hospital could have, and it'll just be in their pharmacy. And then we'll have at advanced cancer centers the more highly personalized CAR cells, made from each person's cells that are could be probably more potent than the ones that would be off the shelf.
Right.
So Carl, for now, you would say it remains pretty much like it is, and at least, let's say, for the next 5 years.
Yeah, I think what we'll see is progress. I mean, there's improvements in manufacturing technology, the media. Like right now, with the cells that Emily had, they had AB negative serum. So that comes from the Red Cross. And when people donate blood, some of that can be saved as plasma or serum. And that we use to help the cells grow. Well, if everyone's getting CAR T cells, there's not enough Red Cross donors then, and for that serum to go around. So it's a critical need to get, the media improved, and the cell culture vessels, and, as I mentioned, automation. So there used to be no one working on this. That's the thing, where there's a science behind it. But no one thought it would ever become a reality. They kind of snickered and said, "What are you doing, making these kind of Frankenstein cells?" And then all of a sudden we found out they worked. And then now an industry is springing up, and there's much work in Europe on this. In fact, people from the automotive industry are now working on cell therapies because they have the expertise in engineering and robotics.
Mm.
Yeah.
No.
We also can see it from our side. Yeah. Okay.
Yeah.
Good. Thank you so much for this comment. I think we still have time to take a few other questions from the audience. So I can see here a question from Lex C. What are your views and experience of the use of gamma delta T cells for oncology? So I guess it's the question to you, Carl, again.
Sure. So gamma delta cells are, they're the kind of cells that Emily got are called alpha beta. So those are T cells that have an alpha beta receptor. There are other set of cells that are less common in your body of T cells called gamma delta T cells. One advantage of them is they don't cause graft versus host disease. So that, if I mentioned earlier to Chris, if I would take my alpha beta T cells and give them to you, unless you're an identical twin, you would get graft versus host disease. And that, in fact, could be lethal. But gamma delta cells, I could take my gamma delta cells and put a CAR into them and give them to someone else, and they would not get graft versus host disease. So there's work on testing those as a way to deliver CAR cells that would have less risk, for the so-called off-the-shelf use. And so those are early stage trials right now, and I'm optimistic. In fact, I think gamma delta T cells might really be very synergistic with alpha beta T cells. There's data from mouse experiments that suggest that.
Right. Thank you. And probably the very, Chris,
maybe
you wanted
one question in between to Emily. Maybe Emily, do you like to share the story how you painted the plasmatherm? Where has this happened? I mean, was it in your room or was it in the kitchen?
I got out all of my stuff and I painted it on my kitchen table.
How long this took you to finish it?
Probably around an hour and a half, maybe.
Oh, great. So it was pretty much the case that we sent over the housing to Emily, and Emily was so kind and was painting. This is the first, let's say, really hand-painted device in the world, which we had, and now it's coated. and it's in our entrance hall here in our factory, and it's an showpiece for all our employees and the team as an inspiration. Thank you again, Emily, for doing this for us.
We still have some of that paint on our kitchen table.
Okay, maybe this becomes part of the showpiece and the masterpiece.
Exactly.
Maybe last question, Olga, from Gonzalo Cabrita. Do you have?
Yeah, I also looked at this one, right?
Do you have any approaches in the pipeline for solid tumors?
Sure. There are, if you look on clinicaltrials.gov and search with the search term chimeric antigen receptor, you would find that there's about 500 trials worldwide. Most of them are in either China or the US. And an increasing number now, maybe 20%, are targeting solid tumors. Some of them brain cancer, some pancreatic cancer. And so that's where the major challenge is. Almost all the blood cancers like leukemia can be treated by CAR T cells in various stages of development. And the field needs to make more potent cells for solid tumors. And so there's a lot of research both in the academic centers and in biotech and pharma now. So, but that is clearly the major scientific challenge to the field.
Yeah, absolutely. Thank you. So I can see another comment. Oh yeah, okay. Thank you so much for your comments, audience. Thank you so much, Carl. Thank you, Tom, and thank you, Emily, for being here and for sharing your story, which is extremely personal and very inspiring for all of us.
Thank you very much.
Likewise, Olga, thank you so much as well from our side. It was extremely a pleasure for us having you guys here today. And I know it's vacation. For Olga and me, it's an ordinary Monday, but you came out. I think you still have an off day, all of you?
Yes.
Yeah.
Wonderful. 4th of July.
4th of July, and so you remain working on July 9th, or when do you start working?
I'm back to work on Wednesday.
Wednesday. For you, Carl, same?
Tomorrow.
Tomorrow, okay.
Yeah. Tuesday.
Then again, from Olga and me, extremely thanks for doing this on your day off here and spending time with us, and of course with the audience around the globe. I would close with some words that we are really deeply touched and inspired. And it's a reminder for us working in a pharmaceutical and life science business here that behind every research, every trial of a breakthrough therapy, there's stories of bravery and dedicated people like you here. And Olga?
Yeah, sure. And also from another side, it also motivates us, the suppliers of the manufacturing solutions for the cell and gene therapy, to accelerate our own development of our own products, to support the new challenges in the field.
Thank you so much, Olga. So we would like to ask you, Carl, Tom, and Emily, if you like, of course, maybe sending out some words of wisdom. This is what we usually do here in our webinar. So, Carl, would you like to start?
I think what we have learned is that science can make major improvements in our health. And over the years, we've had many cuts in funding for research. I think the CAR T-cell story shows why you want to have research, and that the public needs to learn about it through what you and Olga have done to try to broaden this knowledge. And the current COVID pandemic is doing the same thing. We need people desperately realize that we need science research and the benefits that they get from that. So I really think that everyone should realize the importance and then say that this is something that the government should fund. Emily was treated on a trial we had funded not by any drug company, but by the Leukemia and Lymphoma Society. It was philanthropy. So people donating money to foundations like the Emily Whitehead Foundation will make better research and help accelerate, these advances in healthcare.
Thank you so much, Carl,
Thank
for
you.
those words. Tom
Yeah.
and Emily?
Tom?
Well, I think something that I've learned through it all, and it's very frustrating when you do have a child with cancer, when you see how little of the funding goes to pediatrics. But I think this trial that Carl showed that you can actually have breakthroughs in children first that can lead to also helping adults. So it's amazing to us to see that this breakthrough happened, with our daughter first. But now, I would say to our foundation, probably 25 to 30% of the calls we get are from adults looking for help too. But I think it shows that we could put more funding towards the pediatrics because it also does help adults in the end. And from all the families that are out there that are benefiting from what all of you are doing, we'd like to say a big thank you to all of you, because I tell people all the time, when you saved our daughter, you saved our entire family. So you're all part of saving families all over the world, and the patients and the families are very appreciative of everything that's going on, and thankful for all the time and effort that you put into it.
Thank you so much.
Thank you so much for these words. Really touching.
Great.
Okay.
Yeah.
So,
Olga, would you like to send out the last words to everyone?
yeah, so I can't be thankful enough for making part of this edition of Coincides, the Insights with Chris and Olga, the Cell and Gene Talk. Thank you again. Also, I would like to spread out there the message about our dedicated Spotify playlist, where all of our today participants contributed with really nice songs that they like. Emily, Tom, and Carl submitted very nice music and updated our playlist, so don't hesitate to listen and share it. The recorded version of this webcast will be also available, so don't hesitate to share it. And thank you again so much for being here with us.
All right, everybody have a great day.
Bye-bye. Yeah.
Thank you so much.
Bye.
Thank you.
Thank
Stay
you.
safe.
Have a great day. Thank you so much.
Bye.
Bye-bye.
Bye.
Bye.